All right, everybody, it's 1 o'clock, so I think we'll kick off. Thank you, everyone, for joining us today. This is a lunch and learn webinar on integrated pest management. So, my name's Jesse Clune, and this is my colleague Camilla Humphreys, and we're from RMCG, and we've been delivering a larger project about reducing chemical and plastic waste in the cut flower industry for New South Wales. And we've been doing this with support of three incredible industry organisations, Protected Cropping Australia, Wildflowers Australia, and the Flower Growers Group of New South Wales. Before we kick off for the presentations today, I'd just like to quickly acknowledge that Camilla and I were joining from Wurundjeri country down here in Victoria, in Melbourne. And I'd like to extend my respects to elders past and present, and also acknowledge those respects to wherever you're joining from today. So, this project has been funded by the New South Wales Department of Primary Industries and Rural Development, and this is part of the 2021 flood recovery funding, which was co funded by the Australian Government. So, AgriFutures, I guess, conceptualised this project and provided us with the funding, and us from RMCG, we've been delivering a lot of projects, not just this webinar. We can talk a little bit about those towards the end of the webinar.
So I'll just introduce the presenters. For this webinar, we will be looking into the findings from two recent banker plant trials conducted by Dr. Bernardo O'Connor from Biological Services in a gerbera flower crop and myself from RMCG in a capsicum cover crop trial for the Vegetable Industry Soil Wealth Integrated Crop Protection Project. The trials introduce banker plants which are used to host beneficial insects to predate pests. The trials introduce the presentations will discuss how the selection of the right host plants and timing of predatory release can help grow predatory insect populations and reduce key pests like western flower thrip. After the banker plant presentations, we will have Dr. Robert Spoonerhart from Western Sydney University, who will present on how chemical resistance develops and provide you with some practical strategies to reduce the risk of resistance on your farm. And we will then finish with a panel discussion where you can ask the presenters about choosing the right beneficial, timing of release, tips for monitoring, or any other IPM problems you are facing. So please write your questions in the chat and Jesse will moderate. Yeah. So we'll start with Dr. Bernardo O'Connor from Biological Services. Thank you.
Good afternoon, everyone. Can everyone hear me, see our presentation?
You're all good.
Beautiful. I'm Dr. Bernard O'Connor from Biological Services, where I'm an IPM consultant. Today, I'll just be briefly talking about doing IPM in cut flowers, specifically a small trial we did with banker plants in a gerbera crop, and a few new solutions we have for pests like western flower thrips. So, what exactly is integrated pest management? All that it is, it's a strategy of pest control that uses all available tactics to reduce pest populations to acceptable levels, but doing so in a cost effective, environmentally rational, and sustainable manner, something that cannot be done with only using chemical controls. There are many benefits of using IPM. The bugs provide effective pest control 24/7, day and night, and basically even when you're not there. If you use IPM, you'll have healthier plants with better quality produce and have greater quality over longer periods of times compared to when you only use chemicals. This is because every time you apply a chemical to your crop, you wear away at the waxy cuticle on the top side of the leaf, leaving the plant more vulnerable to pests and diseases. If you use IPM, you'll actually spend a lot less time spraying pesticides in your crop. And because you ultimately rely on the beneficials to do the heavy lifting in terms of pest control, you will have fewer chemical resistance issues in the future. This means that your pesticides will actually work as intended when you really need them to. So I'll briefly talk about the banker plant project idea. The idea behind banker plants is to basically provide pollen as a sort of food and a dense habitat for the beneficials. The idea behind them is to provide a favorable habitat to allow the beneficials to establish more quickly in a crop and to remain for longer periods in a crop. If you have a greater number of beneficials in your crop, presumably, and you do have greater pest control. Banker plants are actually a key tool that they use in Europe for the cut flower industry. And we're a little bit behind here in Australia in terms of that. Banker plants are most suitable for Aureus pirate bugs, beneficial bug that predates on thrips, Cucumerus predatory mite that predates on thrips, as well as Californicus, another predatory mite that can predate on thrips, as well as spider mites. In this trial I tested two different species of the banker plants, Sweet Alyssum and a Greek variety of basil. So I'll briefly go over the results of what I found. In the Gerber crop, after we released the Aureus on the banker plants, the Aureus pirate bugs remained in the crop for longer than the previous years when we didn't use the banker plants. In previous years, we released the Aureus and they sort of disappeared after a few months. But when we had the banker plants, it was easy to find the hatchlings, the babies of the Aureus, meaning that the population was sustaining itself very well. It was actually easy to find adult Aureus actively predating on thrips on open Gerbera flowers, something that didn't happen before when we didn't have banker plants. A couple of observations on the banker plant species is that Sweet Alyssum flowered very consistently, whereas basil did not. This meant that Alyssum had more pollen available for the Aureus. And I did find the most Aureus on Sweet Alyssum rather than the Greek basil. So this trial was really qualitative rather than quantitative. There, we didn't have any numbers, solid numbers to compare. It was never really meant to be shared. It was just a sort of industrial rough trial we did. But so you have to keep in mind that there are a few limitations about this. Firstly, is that I used a relatively low number of banker plants in these crops, whereas in Europe they use much higher rates. Presumably, the effect is that the more banker plants there are, the higher the population of Aureus and beneficials you can sustain, and therefore have even better pest control. Also, I didn't actually have a formal controlled treatment in this trial. We only had a couple of greenhouses where we did the same thing, so it's not a fair comparison. Another thing to keep in mind is that these structures where we did the trial were heated overnight. So if you put banker plants and Aureus in an unheated greenhouse, you may not have the same effect. The heat is important for Aureus in order to increase the population quickly. A couple of drawbacks about banker plants themselves is that they can themselves become vulnerable to caterpillars, aphids, and mites and may require chemical intervention to control these pests. And lastly, a little bit of feedback I got from some growers is that the banker plants can themselves become another crop you have to take care of. You have to water it, give it nutrients, and trim it fairly regularly in order to keep the flowering consistent. Speaking of pest control, it's important to be able to find, detect the pest in your crop to know whether your chemical controls or IPM is actually working. So I wanted to share just a few tips on this. Always start in the cleaner, younger crops before going to the dirty areas, because you don't want to go to the dirty areas and bring pests into your cleaner areas. As you walk through your crops, always scan far ahead into the rows on either side and see if you can identify pests or diseases from a distance. Always check several rows in every greenhouse you have, stopping several times at each of those rows and checking several plants at each stop you make. It's incredibly important to inspect plants thoroughly. We always look at the whole leaf and the whole plant, but it's important to look into the growth tips, underside of the leaves, as well as the stems. I strongly recommend using a hand lens to magnify and to detect the presence of eggs or juveniles that are very invisible, pretty much invisible to the naked eye. And always focus on uh, areas that have a little bit of damage because that's where the pest may be hiding. I also like to tell people to look out for, nature gives you a clue. If you see a trail of ants going up a plant, it's more than likely that there will be aphids, whiteflies, or mealybugs there. Always look around fresh signs of damage like chewing, scarring, or scuffing. And if you see on a particular plant damage or speckle the petals on a flower, more than likely the plant is infested with thrips. Also, keep an eye out for things like droppings or sheds in small white, fluffy materials on the plant. So, yeah, the first step to knowing whether your controls are working is to try and find the pests. Lastly, I just wanted to share a couple of new solutions we've got for thrips here in Australia. First off, we've got Franklino thrips. It's a native predatory thrip species. We found it in Queensland a couple of years ago, and we've been using it to effectively control a devastating chili thrips outbreak in Western Australia. Things like Western flower thrips and chili thrips can wreak havoc on several types of crops. So, yeah, we found that Franklin thrips works very well against Western flower thrips as well as chili thrips in combination with other beneficials we've got. And lastly, we've also got the slow release sachets. All that it is, it's basically a small breeding system in a paper bag. And out of a tiny little hole, predatory mites come out consistently for 4 to 6 weeks, and they look on the plants for their pests and hunt them down. The beauty about the slow release sachets is they guarantee even coverage of the beneficials, because much like chemicals, if you don't get good coverage, you will not get good pest control. And that's all for me. Thank you very much.
Very much, Bernardo. It's very interesting to see that the lesson provided a reliable food source due to the long flowering periods and a good habitat for the beneficial oriates. So, yeah, I'd just like to draw a comparison with Bernardo's work in gerbers to a capsicum crop. Um, so, I did this work as part of the Soil Wealth Integrated Crop Protection Project, a vegetable industry levy funded project. And the trial was aimed at assessing the feasibility and benefits of planting alyssum as a host plant for orius and other potential beneficial insects. Myself and biological services representative Anita Merkatz worked closely with a greenhouse capsicum producer, Andrew Browns from Browns Produce in Virginia, South Australia. And the trial aimed to find an IPDM approach to ease Western flower thrip pest pressure in the greenhouse production and to reduce the reliance on spraying. So we installed banker plants, which, as Bernardo explained, act as host plants for beneficial insects. Um, and Aureus was then introduced to predate the problem pest Western flower thrip, which is a significant pest in Virginia as it serves as a vector for tomato spotted wilt virus. So alyssum, due to its long flowering period, was chosen as a pollen source for Aureus and when capsicums were not flowering. So the planting took place last year in August, 2 weeks prior to the first release of Aureus. Bernardo also touched on the density of um, banker plants, and we did a density of roughly 72 plants per 50 square metres. So just bear in mind. So 2 weeks after the second release of Aureus in early October, there were good levels of Aureus hatchlings in the banker plants with low thrip levels. Um, this monitoring data is thanks to biological services who collected the data over the course of the season. Um, and on the 15th of October, spray controls were implemented, including Verses, to control whitefly and dipel for caterpillars, as well as the release of Aphidius for aphid control. So later in the month, October, Aureus population numbers were very good in the banker plants with low thrip levels, whilst whitefly was still very active. And in November, you can see, Bacillus was released at the start of the rows, but you can see that Aureus populations were good in the banker plants. However, myriads were also detected, as well as some thrips. And aphids were controlled with some parasitism and whitefly still around. So this graph shows Western flower thrips and aureus as percentage number of flowers over the time period on the x-axis. And as the aureus predator population was released, there were very good levels of aureus hatchlings from the alyssum banker plants, and at the same time, the number of Western flower thrip went down, and there were no juveniles of Western flower thrip detected. So, yeah, that was a good result at that stage. Later on in the trial, it was actually earlier this year, we dissected the alyssum plants and we did see high levels of mirids and spiders, but they were not found in the capsicum crop, which showed that alyssum also served as a really good trap plant. So the trial demonstrated that banker plants can be used in glasshouses to host beneficial insects and help control pests. However, alyssum was also shown to be a trap plant, attracting other pests, including mirids, two-spotted mites and aphids. So for the next phase of the trial, we're continuing to work with our grower. We'll be looking at new alyssum plants in hanging pots just to see if the height of the banker plants has a beneficial effect on aureus habitation. And yeah, that's a method used in Europe, and we'll just see if it works here. So thanks very much.
Well, thanks so much for that, Camilla. Really interesting to see how the different banker plants have worked, and that alyssum seems to be a good plant across cut flowers and capsicums. So we've touched on some biological control and I guess a way to do it. But I think, Robert, it'd be really great now if you could take us through why it's really important, particularly in the sense of chemical resistance. And I'm really excited to hear what you have to say about that.
Thank you, Jesse, and good afternoon, everyone. Yes, I'm Robert Spurnerhart. I'm from Western Sydney University. I've been involved in IPM in a range of crops for quite a number of years, including biological control. But I've been asked this afternoon to talk specifically about pesticide resistance and how you might manage pesticide resistance. Now, is my image coming up there now, or?
No, not yet.
All right, give me.
Here we go. It's just loading now.
Yeah, now.
Excellent.
Sorry about that. That's the joys of technology. Okay, so I want to start perhaps by saying that pesticide resistance is something which we create. You'll see the definition there. We're talking about the ability in a strain of a particular species. So the strain issue is quite important. We're looking at tolerating doses of toxins that would normally prove lethal to the majority of the individuals in the population. And again, that's an important concept to get across. So if we go back to the very early pesticides, and there's one there on the left, which is sulfur, one of the earliest pesticides ever used, these were quite broad spectrum in the fact that they targeted a whole range of different aspects of pests and diseases. Why is that important? If we move to, I guess, part of the new generation of pesticides, these are now synthetics, and they tend to target either single or specific targets within either insects or fungi. So I want to pick up on that issue a little bit later because I think that's again an important concept to get across. So resistance to pesticides did occur with the earlier pesticides. So even as early as the early 20th century, we were recording some resistance to pesticides. And that image there on the right-hand side is hydrogen cyanide fumigation of citrus trees against scale pests. And this was one of the first recorded cases of resistance. In fact, what the scale pests were doing was essentially stopping breeding so that they weren't fumigated out. As I said, all populations, and that includes humans, but just as well as pathogens and pests, have natural variation. And this is a shot just from one single crop. And you can see here, this is one particular species of fungus, but all of these are separate strains. You can see physically they look different, they're growing different, they're coloured different, because they are genetically different. And genetically different means also that they have mutations associated with resistance to pesticides. So if we take again, this is a very simple, I guess, graphic of what might happen. So at the beginning, when we apply pesticides, there are a number of these individuals, a very small number, and these are here in red, that are resistant. So they carry a resistance gene to a particular pesticide. As we apply the pesticide, so we eliminate the susceptible members of that population. This means that we have a lot more of the resistant ones surviving. The resistant ones then breed up, and then they become the dominant portion of the population. And this is a particular strain, potentially in a particular location. Eventually, as we keep applying, we end up with nothing else but resistant members of that population. So we're talking about populations that can then be moved around the farm or moved to another location or moved, as unfortunately has happened in some cases, between one country and another. So we end up with resistant strains being moved around. What does resistance look like to you in the field? Well, your first indication is going to be that the sprays don't appear to be as effective. The second thing is you're going to need to spray more frequently to achieve the same result. And eventually you'll get to the stage where the sprays don't effectively suppress the pathogens or the pests. So I've referred here to a threshold level. Now, in some crops, we have very clear picture of what threshold levels are. But in the case of ornamentals and cut flowers, basically any damage is damage, and that's the threshold. So we have an aesthetic threshold, which is much lower than it is for most other crops. And again, that's very, very important in terms of resistance. So what types of resistance mechanisms occur in both insects and also fungi? Well, there are two broad types, I guess. There are those that induce a fairly low level resistance. So we might talk about 10 times, 15 times, 20 times resistance. We call that relatively low. This is where we get a decreased penetration or uptake. So, for example, we might get a thicker cuticle on an insect. We might get a thicker structure on a fungus, which slows down the rate of uptake of the pesticide. We can also get behavioural resistance. But again, give you, for example, an insect which can move to the undersides of the leaf or be in a more protected spot. And then these are much less likely to be affected by pesticides. But these are what we call low-level resistance, and these aren't normally the ones that result in a breakdown of a product. There are then two that are associated with what we call high levels of resistance, and these can be in the order of hundreds of times or more resistance factors. So anything which would normally kill, you now need 100 times that amount to achieve the same result, and it basically becomes unworkable. And these are two that I've mentioned here. The first one is a detoxification mechanism, so that the pesticide is actually detoxified, so it doesn't function as a pesticide. The second one is that the target site for which the pesticide has been developed is no longer sensitive to the pesticide. And here, as I say, we get high level of resistance, and these can lead to breakdown of effective pesticide use in the field. Now, I'm also going to mention the term cross resistance because I think this is important. Pesticides have the same mode of action. If resistance occurs to one of those, it could also confer resistance to others of those within that same mode of action group, even though they've never been exposed to them. So becomes very important when we start to look at how we select which particular pesticides we are going to use to make sure we minimise resistance buildup. The other thing is, if pesticides are then removed, what happens to that population? Does it kind of revert back or does it stay resistant? And both situations could occur. It depends on whether or not those genes are actually stable and the mutations are stable or whether they break down again with time. But certainly we can consider that in many cases, once a strain is resistant, it remains resistant. So what are these mode of actions that I'm talking about? Well, there are a huge number. So for insecticides, there are more than 36 different modes of action that have been documented. And for fungicides, there are a lot more. If you want to have a look at where this information is available, my suggestion is you go to CropLife Australia. And these are two tables. The one on the left-hand side is the front page of the insecticide, and the right-hand side is the one for fungicide activity. So you can actually look up all of these particular fungicide activity groups. You can also get exactly the same information off each label. So you'll see here on the left-hand side, we have a group 5 insecticide, which is a spinosyn. That's the group, and it has a particular mode of action. On the right-hand side, we have a group 11, which is a strobilurin, and again, it has a particular mode of action. So each one of these has specific modes of action, and there are potentially a number of pesticides in each of those groups. But on the label of every one of these, you will see that information as to which group they belong to. Now, Bernardo's picked up on a number of these issues, so I'll go through some of them fairly quickly. But what are the strategies to minimise pesticide resistance? And the first one, of course, is to adopt IPM strategies, or integrated pest and disease management strategies. And again, Bernardo's talked a lot about monitoring. The only thing I want to add to his comments is that for ornamentals and cut flowers, it is absolutely critically important because we're looking at essentially picking up at the first stage. In other crops, we may be able to look at some sort of a nice balance. But in the case of cut flowers, often we can't wait. We don't want that sort of balance. We basically want to get in as soon as we possibly can. We have cultural managements, and these includes things like hygiene and plant sanitation, quarantine and biosecurity, maintaining good plant health. So irrigation, nutrition is a critically important component of ensuring that you get minimal pest and disease damage, particularly in protected cropping. So you'll see a whole list of these down here, all of which are associated with what I will call that cultural management, which is really the prevention of pests and diseases getting a hold. We then, of course, have biological control, and Bernardo's company is one of the two major companies in Australia supplying biological control agents. So I won't dwell on these at all. You can go to the Good Bugs website, which also lists a whole range of essentially all of the biologicals that are available against all of the pests that are currently in Australia. There's also a biological control of plant pathogens. So just be aware that there are a number of products that are biological controls for plant pathogens as well, as an alternative to synthetic fungicides. There are physical and mechanical methods also, and I particularly want to highlight the mass trapping down the bottom there. You've got the blue sticky, which is used for mass trapping as well as for obviously the yellow sticky and the blue sticky traps that are commonly used for monitoring. So now let's look at, we're using pesticides. So what are the strategies that we can adopt to minimise pesticide resistance? And we're really looking here now at adopting stewardship or resistance management practices. So the first of these is make sure that you know that you've correctly identified the pest or the disease. And unfortunately, I guess I've seen, and I guess all of you have seen too, situations where there's been misidentification and applications of pesticides have been made that are incorrect. The second one is to minimise it, optimally time your pesticide applications. The third is to target the most vulnerable stage of the pest and disease life cycle. And this is very clear in some cases where we know which is the most critical stage of which to apply the pesticide. Rather than spraying the entire crop, you spot and target apply pesticide. This means that you're not applying across the entire crop. It's not just an economic thing, it's also resistance management, and it also is environmentally the right thing to do. Ensure that we have good pesticide coverage, and this is particularly so for non-systemic pesticides. Select wherever you can non-disruptive pesticides when you're using biological control. Make sure that you follow the directions on the label regarding the frequency per season and the spray interval times, because this is all part of the resistance management strategy that's been developed for the product. And rotate the pesticide groups used, adhering to the advice on the label. And for example, here's the label for one of the strobilurins. You'll see it talks here about applying in sufficient volume to make sure you get adequate coverage, but also applying a maximum of 2 consecutive applications with a minimum treatment interval of 14 days. So that specific information is available for you on the pesticide labels. There are also a range now of multi-pesticides, and you'll see here there are 2 different products together from 2 separate groups put together in the insecticide on the left and on the fungicide on the right. So this is a strategy that's being adopted by the pesticide companies to also minimise pesticide resistance buildup by incorporating 2 products of different modes of action in the same product. So I'm going to finish off by saying if you think that you have pesticide resistance, you really do need to check that out. And so contact your consultant, contact your pesticide supplier, or contact New South Wales Department of Primary Industries, or if you're in another state, your local Department of Primary Industries or Department of Agriculture. And my last takeaway is if we're looking at the key cut flower pests and diseases that have resistant strains, two-spotted spider mite, western flower thrips you've heard about, green peach aphid, cotton aphid, silverleaf whitefly, serpentine leaf miner, and for the diseases botrytis or grey mould, myrtle rust, and powdery mildew. So with that, I will acknowledge the funding for this work, and I also have some questions perhaps that people might want to ask. Thanks very much.
That's excellent, Robert. Thank you so much for that in-depth coverage of chemical resistance and kind of how it develops. So while we're waiting for some chats to come through, just a quick bit of housekeeping. Ben, yes, we are using the Q&A function. Sorry about that. So if anyone's got any questions for our presenters, chuck them into the Q&A function. That should just be at the bottom of your Zoom screen. If you can't see it, just jiggle your mouse across your Zoom screen and it should come up and it'll just be the Q&A function there. But Camilla, I believe you had a question to kick us off.
Thanks, Jesse. No, thank you very much, Robert, for your presentation, and Bernardo. I guess what you were talking about, Robert, was once resistant, always resistant, and the target site can no longer sometimes is no longer sensitive to the insecticide, and so therefore you need to continue to rotate your mode of action groups. And she showed us the crop life um, website, which people can access to look at the different modes of action groups. You emphasise the fact that it's really important to prolong um, the effectiveness of active groups, and particularly, well, Group A, 28 amongst others, and Group 28 I was interested to ask you more about because it's a softer mode of action group chemical. And yeah, my question was, how are we going to continue to be able to use mode of action group with those things that were discussed in your presentation?
Okay, thanks for that. Yes, the group 28's I think they're the ryanodine receptor group. And yeah, look, I guess one of the first things is that if you look across particularly the insecticides, the vast majority of them probably target various parts of the nervous system. And that's so in this particular case too, even though some of these, as you say, are what we refer to as softer chemicals. You've still got the same issue, the fact that it is a very specific target site. Um, these have been sometimes that's the way that they've been discovered because people will actually look at certain target sites and say, what are the chemicals that are effective against those target sites? Um, yes, the fact that they are, I guess, safe relatively safe in terms of narrow mode of action. But the other side of it is that anything that is specific, you are going to have resistance ultimately develop, particularly if you continue to overuse them. So I guess one of the things, yes, is to look at rotating your chemical groups. And yes, they're not always those resistant populations aren't always stable. I've spoken to a number of colleagues who do work in this area, particularly the resistance monitoring work, and they've told me that they've actually had some of their strains that they've been keeping for a number of years that have now lost resistance. So it is actually possible that if they've been not exposed, and so you don't get that selection pressure all the time, that maybe there are characteristics within them that tend to enable them to go back to a degree of equilibrium. So that's certainly one of the things which I would recommend. But I think the most important takeaway is pesticides can be used effectively strategically as part of an IPM program, but they're not your go-to chemical. I mean, they're not your go-to mode of action. They're not your go-to control. You really need to be looking at integrated pest management and utilising biologicals wherever you possibly can.
That's excellent. Thank you for that, Robert. And Sarah, just quickly before we get to your question, just a quick follow up on that one. Um, why is it actually important that we protect these chemistries we implement IPM? Are we not just going to come out with new chemistries over the years?
Well, I guess look, let's take the salient problem that we currently have with antibiotic resistance. Okay, I guess most people are aware that we have multi-resistant golden staph. We have um, basically no new antibiotics coming out on the market. The same thing's happening with pesticides. I mean, the number of different pesticides, particularly new pesticide groups that are coming out, are very few and far between. So we really have to make sure that whatever we have in our arsenal at the moment can last as long as possible. And look, going back a number of years ago, I recall one particular product that was used for two-spotted spider mite. It lasted 2 years, 2 years in the field, and then it was gone. So yeah, we just can't rely on people using pesticides in that way any longer. We know about it when we think about, as I say, what happens with human pathogens. So the story's been going actually quite a lot longer with particularly insect pest resistance. So, yeah, there is really nothing new. If there are, they're going to be few and far between, and we still got to have to make sure that we manage all of those effectively too.
That's excellent. Thank you so much, Robert Openadic.
Just to add to Robert's point, I was going to say the exact same thing. A new product came out for wildlife control in open field tomatoes, eggplants, and all these things. 2 years later, we're starting to see the chemical not work anymore. It was day and night. From the month it came out, it just cleaned up beautifully. 2 years later, we're lucky to get 25% knockdown. So it's exactly as you said. The brand new chemicals will stop working.
That's excellent. Thank you so much both for that. Now, I think going back to the banker plant stuff, Sarah Scott has asked if you can use these banker plants outside in the field, or is it a strategy that's better for greenhouse environments?
So I would say the banker plants are definitely applicable in open fields, and really that's what we're trying to mimic: being in a natural open field where you've got a bunch of flowers and habitats for different bugs, and to achieve that proper equilibrium of enough predators and enough prey so that nothing gets out of control. So there are many crops where people are trying to plant at the sides of their crop, plants that are consistently producing pollen and flowers, precisely to harbor a diversity of different predators to prevent any one pest to get out of control.
Can I perhaps add the comment there? In the case of protected cropping, what we're looking at are the biologicals that we've released in there. In the field, we're also looking at the potential of naturally occurring biologicals. And so you're actually broadening the base of the biologicals that could be available in the field situation. Um, can I just make one other comment? And I'm interested in Bernardo's response. Um, we've been doing a little bit of work recently, and this is not specifically in the cut flower area, but we've been looking at the issue of pollinators in greenhouse cropping and the use of, for example, banker plants to actually support pollination activity in protected cropping. So, just wondering whether you've had any experience with that. Our experiences have been very good, by the way. Um, so we've been looking at flies, we've been looking at stingless bees, for example, um, and basically providing them with an alternative resource to the crop. So, have you had any experience at all in that area, Bernardo?
Um, I haven't seen much. What I've seen is some people introducing beehives into their greenhouses or glass houses, but they were affected by pesticide applications, of course, and they didn't seem to navigate very well because of the way the light diffracted from the roof
Honey
and all
bees
of that.
don't. No.
No, so they didn't do too well. But I did think the same thing, Robert. Flies have got to be contributing, blowflies as well. They're a little bit, you don't find them too many in crops where you'd like to see them, like greenhouse strawberries, greenhouse tomatoes. Um, that is something that we're struggling with in Australia. We don't have bumblebees like they do in Europe, which do a fantastic job. Um, and yeah, unfortunately, I don't have much to contribute. Just you kind of hope flies come in and enough of them to help you out. But mostly where I work, it's done by hand or by back blowers and things like that.
Okay, thanks. Yep.
Cool. That's excellent. And while we're on the topic of banker plants, Ben from Wildflowers Australia is curious. I think this was one for you, Camilla, but Robert and Bernardo, if you can jump in this one as well. You said that there was some spiders that came into your alyssum plants. Are they going to eat the beneficials?
Um, Bernardo is an expert in this area. We didn't go on to monitor to see if these spiders were eating the beneficials. Um, so I can't answer you directly, Ben. Apologies. But that's certainly something to look into for the next trial. Bernardo?
If I could, yeah, if I could just add to that, it's extremely rare. There was one particular situation, one single farm where spiders became a bit of an issue. They were eating a lot of Oreos, but again, it's one out of 200 farms that we've got in South Australia where this is actually happening. It could be a multitude of things that could also be affecting the Oreos, and the fact there are a few spiders around maybe contributing, but we just don't know. Again, it's not a concern for the average grower out there that there's too many spiders eating my Oreos and therefore I have thrips damage or tomato spotted wilt virus or other things coming in because of lack of thrips control. It's extremely rare.
That's excellent. And I guess kind of on that theme of, I guess, some of these complexities you see with the banker plants, so obviously spiders aren't going to be eating all your beneficials, but are there things that growers should be considering when they're implementing banker plants into their existing production system?
I think I would keep a few things in mind. First, put enough of them in. If you just put a couple of hanging pots per hoop or per house, however you want to call it, you can hope to have much of an effect. It's something like we did in the trial I did. We had only very few. Like I say, in Europe, there's rows of them for every hoop, for every house, however you want to call it. Um, so that, of course, gives you the most amount of habitat, the most amount of pollen, and for the most chance to actually get them established and maintained in your crop, providing with effective pest control. I would also suggest use something that's easy to maintain. Alyssum seemed great. It didn't overgrow. It consistently flowered, and it wasn't too thirsty or wasn't too demanding in terms of fertilizer or anything else. So something that's easy, something that's not going to add too much work to give you the most benefit.
Mm, No, definitely, definitely. And I imagine when you're installing your banker plants into your cropping system as well, you'll have to do that monitoring to make sure that you're attracting the right things in as well. Continue on the banker plants. Sarah was wondering if there's any native flowers or perhaps some native grasses that would be a suitable banker plant. Camilla, I know you looked at some native insectaries as part of your work for soil wealth.
Yes. Um, yeah, I won't discuss that. That was unfortunately decimated by herbicide. So we didn't even round up monitoring that trial, unfortunately. But yes, Bernardo or Robert, if you want to level.
I can't think of any species names in particular, but as a rule of thumb, I would say anything that, as long as you maintain a complement of plants that flower at different times. One that flowers, I don't know, January through March, another that flowers March to July, and so on. So there's always a little bit of pollen being provided throughout the year. I know there's a certain variety, a certain assemblage of native plants you can use, easy to maintain, that will give you just that. I am sure if you do a quick Google search, you will be able to find a list somewhere. I'm pretty sure I've seen something like this.
Oh, that's really, really excellent. And I guess maybe we'll circle back a bit to the pesticides, or I guess we both talked about thresholds. But Robert and Bernardo, I'd love to hear your thoughts about growers establishing their own thresholds in their crop. What should they be thinking about? Can they Google thresholds for roses, or is it something they have to do themselves?
Um, I have my own view on grower thresholds, and it is that you have to sleep at night. Uh, if you're going to bed worried that should I be doing something or should I not be doing something, in terms of a threshold, then I think that's when you probably hit your threshold. And I don't mean that in a difficult way. I'm saying particularly with cut flowers. What you can tolerate out of season when you're actually assuming you've got particular periods of time when you're actively cutting flowers, then the rest of the time you can obviously tolerate. You have a much higher threshold because it's not in a reproductive phase. Um, the same thing is in the case of things like roses, and it occurs in a whole range of other crops. What happens towards the bottom of the canopy? You can have quite an integrated system taking place down there with perhaps a few pests and a few beneficials. But as long as they're not getting up to the productive part, then you don't have a threshold issue. So I think it's really important that you look at that in a context way. I mean, I've seen cases where, for example, there's been, and I'll give one example in wildflowers is scale pests, where you will have some scale pests. You will start to see some predation or some parasitism occurring. You'll be able to see that because you'll see holes in the scales, or you'll see maybe um, some immature stages or even adult ladybirds crawling around so that you know you've got those there. But as long as those scales aren't getting out of hand and getting up into your cut flower area, which is where the issue is, then most growers are now learning that they can tolerate that at that sort of a level. And if you don't, then you really don't have a nice biological system in place. And I don't think people want to be overusing pesticides just for the sake of overusing them. So that would be my take on it. But I do agree that particularly because you're out there all the time, once you're cutting, maybe you're out there cutting every day or every couple of days. So you're actually kind of monitoring the process while you're out there as well. So, but that means that you need to take aware of, I guess, the productive part and the maintenance part of your plant. Does that make sense?
No,
Yep.
that was great. Bernardo, did you have anything to add to that one?
I think Robert hit all the important points. I think the average grower really knows because, again, like he said, you're out there cutting your product, you're looking at it, and a very good grower just by one quick look can tell, okay, this is a problem. This happened a few weeks ago. What happened a few weeks ago? This pest came in. Didn't do this spray. They've got a very good gauge already. It's all good and well to say, oh, theoretically, when the trips are 20% on 20% of the leaves after I monitored, that's fine to do. But really, if you just walk in and you know that things are not right and you need to do something about it, I think a good grower knows. Yeah.
That's excellent. And Robert? That's excellent. We've got a question from Ben. So when your bank plants get full of perhaps not so beneficial things, do you spray them and then reintroduce your beneficials, or is there another strategy you can use here?
Maybe Ben's talking specifically about me saying that the bankers got caterpillars, aphids, or mites. Um, for example, where I was doing this trial, the alyssum plants got smashed by diamondback moth. Um, and yeah, all the grubs ate a lot of the foliage, and they didn't look so flashy anymore. Lost quite a few flowers. But just a simple application of group 28 chemical like what's it called collagen or belt, something like that, uh, would not interfere with aureus at all and would be very effective at controlling the caterpillars. And no problem, control the caterpillars, you don't harm the aureus. If you use something a lot harsher, yeah, you would be affecting the aureus. But again, like Robert said, there are a lot of the chemicals used in IPM are very selective, that they'll very specifically target one pest's, metabolic pathways. They'll only affect that one, not the aureus. There's always a little bit of a side effect, but usually, again, we try to use what's friendliest to the beneficiaries. So there are options.
And can I just add, if you are using biologicals, then the company that's applying the biologicals will be able to provide you with all of the information in regard to what particular pesticides you can use and over what period of time should you not be using those pesticides, either prior to or immediately after application. So a lot of that information is known, and all the biological control suppliers will certainly assist you in that process.
That's excellent. And one last quick question before we wrap up today's webinar. What's a take home action from both of you that growers can do today, either in regards to managing their chemical resistance or their wider IPM plan?
If I get in first, I'll beat Bernardo because he'll probably say the same thing. And that is effectively monitor your crop. I mean, monitoring is the key. That's the thing that is, you're going to be out there, you're going to be in charge, you're going to know what's going on, not only in terms of when you might need to do something, but it's also, as Bernardo was saying, after you've applied something, you need to be monitoring to see whether it was effective or not. So to me, that would be my number one takeaway message. Monitoring is the key to any effective pest management.
Excellent. And Bernardo, Robert, still your take home message?
Yeah, one of the key ones, absolutely. That's essential. Good monitoring. Um, I think if a second key takeaway would be do use the beneficials. It may seem like I'm pitching it, yes, buy from my company, but it's beyond that, really. Um, there are very few new chemicals coming out. New chemicals eventually stop working, and the only thing that you can use in between those sprays is the bugs. A lot of people think it's too expensive or too new a technology. It's been around for decades and decades and decades. It's proven, it gives you a break, and they really do a job.
And I'll just, my final take home comment, not message, is we don't know yet of too many insect pests that have become resistant to predators.
Oh, that's lucky. Lucky for us. Everyone who is at home, please join me in extending a really warm thank you to Robert and Bernardo and Camilla for presenting their work on vapor plants, resistance, and sharing their wisdom around IPM. And just before we leave, I've got a shameless pitch. So we have three workshops coming up on integrated pest and disease management. These will be in-person workshops, and they will be up in New South Wales, in the Central Coast, the Far North Coast, and the Mid North Coast. And they're completely free for cut flower growers like yourself. So if this is something that you're interested in coming along to, scan one of the QR codes that you're seeing on the screen now. If you can't get through, have a Google, it'll come up, or you can send me an email at jessec@rmcg.com.au and I can point you in the right directions. Again, thank you so much, Robert, Camilla, and Bernardo, for your work today. And I think we will hopefully all take home a good IPM practice, whether that be introducing beneficials into our system, making sure that we're really monitoring, and of course, make sure we're rotating and only using our chemistry sparingly because we're not going to get any more. Thank you all and enjoy the rest of your day.